Welcome to the Deep Dive. Ready to plunge into bioinformatics? We've got a stack of articles hot off the press, covering everything from sequencing to AI and rare diseases.
It's really exploding with new discoveries. Hard to keep up.
That's why we're here, to break it down. So, first up, direct RNA sequencing, or DRS. It's shaking things up.
PRS, yeah, it's a game changer. Scientists used to have to convert RNA to cDNA before sequencing, like translating a book before reading it.
Adding an extra step and maybe some errors.
Exactly. But DRS lets us study RNA directly, a clearer, more accurate picture.
We've got an article on a new tool for DRS, SeqTagger. It tackles demultiplexing, a major bottleneck.
Imagine a jumbled pile of letters all mixed up. That's demultiplexing. SeqTagger uses an algorithm to separate the data rapidly and accurately.
Like a high-speed sorting machine, making DRS more efficient.
It is, and more reliable too, opening up possibilities for research and drug discovery.
DRS is the future, but we can't forget the past.
Yeah.
All that info in formalin-fixed, paraffin-embedded tissue samples, the FFPE archives.
Yes, FFPE samples. A gold mine, but tricky.
Right. They're often all we have for retrospective studies, but the preservation can degrade DNA and RNA. Makes it hard to analyze with next-generation sequencing, or NGS.
Like piecing together a fragmented scroll, you need the right tools.
One article stresses choosing the right library preparation kit for NGS with FFPE samples. It's not one size fits all.
No, it's not. Different kits are designed to handle the challenges of FFPE. Some are better with degraded material. Others are optimized for low-input DNA or RNA. Researchers need to really consider their samples and goals.
Matching the tool to the task.
Precisely. Unlocking the potential of FFPE samples hinges on that.
Let's shift to liver cancer, where bioinformatics is having an impact. A study from the Karolinska Institute.
They used RNA sequencing to look at the immune response to liver cancer. What they found is really exciting for immunotherapy.
Specific T cells in the liver and lymph nodes that show promise in fighting it.
Yes, a huge step towards more effective, targeted immunotherapies for liver cancer. It's notoriously difficult to treat. This level of analysis of the immune response is only possible because of advances in RNA sequencing and bioinformatics.
It's like having a battle map, pinpointing weaknesses and being more strategic.
And that precision is key in the fight against cancer.
Bioinformatics is also making a difference in rare diseases.
Those often present a diagnostic puzzle and go undiagnosed for years. Advanced sequencing technologies come in here, like PacBio's synchronized FiberSeq and Kinnex, formerly MASSEq.
Like looking at a rare disease with a high-powered microscope. Seeing not just the genetic code, but how it's expressed and regulated.
It unravels the complex basis of these diseases, opening doors to treatments and deeper understanding.
One article shows how this helped untangle a tricky, rare Mendelian condition.
Providing a fuller picture, these techniques are empowering researchers and offering hope to patients and families. A powerful example of bioinformatics transforming how we approach rare diseases.
Remarkable. But there are challenges with a field advancing this quickly, right?
Of course. One article asked, what happens when the AI revolution, which relies on data, runs out of data?
We overlook that sometimes, don't we? We're generating tons of data, but the demand for high-quality, labeled data for AI training is huge.
Data scarcity could become a roadblock, especially in bioinformatics where AI is so important.
Like building a rocket but running out of fuel before orbit.
That's it. The article points out some strategies like synthetic data and federated learning, where models are trained on datasets without sharing sensitive information.
Even with these advancements, we need to be mindful and look for solutions.
Exactly. Innovation means anticipating challenges before they become huge obstacles.
We've covered so much, from direct RNA sequencing to working with FFPE samples and the promise of T-cell therapy. We even talked about advanced sequencing for rare diseases and data scarcity in AI.
And that's just the start. In the next part, we're going deeper into AI and its applications in bioinformatics.
Stay tuned. We'll be back.
Welcome back to the Deep Dive. Before the break, we talked about sequencing and its impact on disease research. Now let's look at another big player in bioinformatics, AI.
AI is everywhere these days. It's making waves in bioinformatics too, unsurprisingly. We've got a bunch of articles that show just how powerful it can be.
One that really stood out to me, it talks about prompt injection attacks. It's a vulnerability in vision language models, particularly those being used in oncology.
Sounds worrying. Can you break that down a little?
So imagine an AI that analyzes medical images, like scans to find cancer. A prompt injection attack tricks the model into misinterpreting the image by injecting these malicious prompts or instructions.
So instead of identifying a tumor, the AI could see something that's not there, or omit something that is, that could really affect diagnosis and treatment.
Exactly. It shows us a crucial side of AI we can't ignore: security. We're giving these models sensitive medical data, so we need to make sure they can't be manipulated.
It's like any powerful technology, right? Potential for misuse, whether it's intentional or not. We need to address these vulnerabilities, especially as AI becomes more common in healthcare.
Absolutely. Cybersecurity in healthcare is so important. But moving on, another article highlights a different use of AI in genomics: genotype imputation.
Genotype imputation. Remind me what that is again.
It's like filling in the blanks in a genetic puzzle. We don't always have complete genetic information for everyone. Genotype imputation uses statistics to predict the missing genetic data based on, data from similar people.
Like using context clues to fill in missing words.
Exactly. The article talks about a new method, STCI, split transformer with integrated convolutions. It's a deep learning model, and it's supposed to make genotype imputation more accurate and efficient.
And why is accuracy in genotype imputation so important?
It's crucial for a lot of reasons. For researchers, it means they can work with larger, more complete data sets, which increases the power of their analysis.
So we can get more insights from the data we have, maybe lead to new discoveries about genetic traits and diseases.
Right. A great example of AI enhancing our ability to understand genetic data.
Some AI applications are about filling in those gaps, and some are pushing the boundaries of what we can do with the data we've already got.
One article talks about a tool, MetaQ. It's designed for analyzing single-cell data.
Single-cell data analysis is a hot topic these days. What makes it so interesting?
Well, it lets us understand cellular heterogeneity. Before, we studied cells in bulk. We averaged out the differences. But with single-cell analysis, we can zero in on individual cells, see how their gene expression, protein production, all that varies.
Like looking at a forest and then zooming in on the individual trees, seeing how they're different.
That's a good way to put it. MetaQ uses quantization to analyze single-cell data quickly and accurately, so researchers can see those subtle differences between cells, which can lead to insights about disease and therapeutic targets.
Like having a really high-resolution map of the cells, revealing things we've never seen.
And those details are important for developing treatments.
Let's talk about AI in clinical notes. There's an article about using GPT models to de-identify clinical notes.
De-identification is key to protecting patient privacy, especially as we share and analyze more data in healthcare.
Right, we want to learn from the data, but keep people's identities safe.
Exactly. De-identification has always been a manual process, really time-consuming, often relying on humans to redact information. GPT models, which are large language models, are showing a lot of promise in automating this process.
So AI can do the heavy lifting instead of humans, going through pages and pages of notes.
That's the idea. GPT models are trained to find and redact names, addresses, dates of birth, that kind of thing, all while keeping the important clinical content.
That could save a lot of time, so healthcare workers can focus on caring for patients.
It could. And make data sharing more secure and efficient, which means faster research and better outcomes for patients.
Everybody wins. Moving from language to images, there's an article about using biologically inspired graphs to explore massive genetic datasets.
A fascinating example of borrowing from nature to solve tough computational problems.
How do these graphs actually work?
Well, think of a spider web and how all the threads are connected. Biologically inspired graphs mimic this to show complex biological systems, like gene regulatory networks or how proteins interact.
Instead of seeing genes by themselves, we can see how they relate and influence each other.
Exactly. These graphs help researchers visualize and analyze genetic data in a way that makes more sense. It's easier to find patterns and relationships that way.
It's like using a map to navigate a city instead of just wandering around.
That's a good analogy. These maps are helping us understand biological systems, leading to new insights about disease and potential treatments.
Shifting gears to a medical application of AI, there's an article on diagnostic reasoning. It highlights a medical large language model that can diagnose across different specialties.
This is a big development, a model that looks at a patient's symptoms, history, lab results, even genetics, to suggest diagnoses across many medical specialties.
Like having a whole team of specialists right there to help with diagnosis, even the really complicated cases.
That's the goal, and we're getting closer. These models are still being developed, but they could really improve diagnosis, especially where access to specialists is limited.
A game changer for patients in rural areas or underserved communities.
It could be. democratizing access to that kind of healthcare expertise.
Some AI applications focus on replicating human skills. Others are creating entirely new approaches.
One article goes into a pretty complex example. Chaotic gradient-based optimization with fuzzy temporal optimized CNN for predicting heart failure.
Break that down for me. What's so special about this approach?
I know it's a mouthful. Basically, it uses these advanced computational techniques like fuzzy logic and convolutional neural networks to analyze medical data and predict heart failure.
So it's not just relying on the usual risk factors. This model is digging deeper, looking for patterns that suggest an increased risk.
That's it, like a super sensitive early warning system.
That could allow for earlier intervention, maybe prevent heart failure completely.
Exactly. This shows how AI can find patterns and predict things that traditional methods might miss.
Some AI is super complex. Others show that simple data analysis can be powerful too.
One article looks at using machine learning to predict what happens after transcarotid artery revascularization, a procedure for carotid artery disease.
What does that procedure involve?
The carotid arteries in your neck supply blood to the brain. Transcarotid artery revascularization is a way to open up blockages in those arteries. Minimally invasive, too.
So it's clearing a blockage to get blood flowing to the brain again.
Yes. The study used machine learning to look at data from patients who had this procedure, trying to find factors that predicted their long-term outcomes.
So they could give personalized predictions for each patient instead of just using general statistics.
Exactly. This personalized prediction can help doctors make better decisions about treatment. And patients can understand their prognosis better.
A great example of how AI can be used to really improve patient care.
It is, and it highlights the importance of using these tools with real clinical data to improve the quality of care.
Moving from arteries to cells, one article talks about an advance in structured illumination microscopy.
This type of microscopy lets us see tiny details inside cells. But the images have always had a lot of noise.
Noise. What does that mean?
Imagine trying to hear a whisper in a loud room. The noise makes it hard to hear what's being said. Noise in microscopy is those random fluctuations in the image. They hide the details.
Like static on a TV screen, making it hard to see the picture.
Exactly. This article talks about a new way to model the noise so it can be removed. So we get higher resolution images.
Like using noise-canceling headphones so you can actually hear the whisper.
Exactly. This advance is pushing the limits of what we can see, microscopically, opening up new possibilities for studying cells.
A reminder that there's always room for innovation, even in a field like microscopy.
Absolutely. And those innovations often lead to big breakthroughs in other areas, too.
Some research looks at visualizing the microscopic world. Others are trying to identify and classify the tiny organisms living there.
One article talks about a new MALDI-TOF mass spectrometry database. It's for identifying and classifying highly pathogenic bacteria.
MALDI-TOF mass spectrometry. What is that exactly?
It uses lasers to analyze the mass of molecules. It creates a unique fingerprint for different substances.
Like using fingerprints to identify people, but on a molecular level.
Exactly. In this case, they made a database of fingerprints for lots of highly pathogenic bacteria. The ones that cause serious illness.
Sounds like a useful tool for public health and infectious disease control.
It is. It lets researchers and clinicians quickly identify potential threats, and then they can take steps to stop the spread of infection.
Like a rapid response team, they can find the culprit before an outbreak happens.
That's a great way to think about it. That quick identification is key to controlling infectious diseases, especially with antibiotic resistance growing.
Let's move on to chronic diseases, particularly type 2 diabetes.
Type 2 diabetes is a complicated disease. It involves genes and environment. One article looks at using advanced analysis methods to find individualized dynamic network biomarkers for type 2 diabetes.
Individualized dynamic network biomarkers. That's a mouthful.
It is complicated, but it's also really powerful. This approach looks at how genes and proteins interact with each other over time, forming these networks.
So it's like mapping out the social network of molecules involved in diabetes, seeing how they influence each other and how those connections change.
That's it. And researchers can use those networks to find patterns, biomarkers that are unique to people with type 2 diabetes.
That could be more personalized treatment, right? Targeting the specific pathways that aren't working right in a patient.
That's the goal, moving away from the one-size-fits-all approach and towards strategies that are tailored to the individual's biology.
The future of healthcare, taking into account everyone's unique situation to come up with more effective treatments.
Absolutely, and bioinformatics is making that happen.
Now let's talk about drug discovery. Bioinformatics is having a big impact there, too.
Traditional drug discovery is slow and expensive, a lot of trial and error. Bioinformatics is helping to speed that up and make it more likely to succeed.
One article shows a cool example of this, using molecular dynamic modeling to look at new drugs from marine seaweed metabolites for cervical cancer,
Marine organisms are a great source of compounds that could have therapeutic benefits.
like searching for medicine in the ocean.
Right. Molecular dynamic modeling lets researchers simulate how these compounds interact with molecules in the body, things like proteins involved in cancer.
Instead of testing tons of compounds in a lab, they can use simulations to narrow it down to the ones that are most promising.
It's way more efficient and cheaper. It's helping researchers find new treatments for all kinds of diseases.
It's amazing that we can use computers to design drugs now.
It really is. Shows how bioinformatics is changing the pharmaceutical world.
While some are looking for new drugs, others are trying to understand how diseases work, especially the ones involving cell death.
Cell death is a normal process. It's crucial for life, but when it goes wrong, it can cause a lot of different diseases.
There's an article about ferroptosis. It's a specific type of cell death, and they're looking at its role in IgA nephropathy, a kidney disease.
IgA nephropathy is when the body attacks its own kidneys, causing inflammation and damage. Ferroptosis is a kind of cell death that involves iron-dependent lipid peroxidation.
Could you explain that in simpler terms?
Think about how iron rusts. Ferroptosis is similar, but it happens inside the cell, damaging the membrane and killing the cell.
So it's like rusting on a cellular level.
Exactly. The study found that ferroptosis is a big part of IgA nephropathy getting worse. This suggests that targeting this pathway could be a good way to treat it.
So if we understand the mechanics of cell death, we can create more targeted and effective treatments.
Right. It shows how important basic research is. Figuring out what causes a disease can pave the way for new treatments.
Some research focuses on understanding diseases. Others are trying to design new molecules with therapeutic potential.
One article talks about the de novo design of protein mini-binder agonists of TLR3.
De novo design. Sounds futuristic.
It's becoming reality. It means creating completely new molecules from scratch, using computers. In this case, they designed protein mini-binders that activate TLR3.
TLR3. What is that?
It's a toll-like receptor, a protein that's a big part of the immune system. It recognizes and responds to threats like viruses and bacteria.
So these mini binders are like specially made keys that can unlock specific parts of the immune system.
That's a great way to think about it. This could lead to new treatments for infectious diseases, cancer, even autoimmune disorders.
A powerful example of bioinformatics being used to come up with solutions to complex medical problems.
It really shows how biology, chemistry, and computer science are coming together in drug discovery.
Let's talk about how AI is changing image analysis.
One article compares different deep transfer learning models. They're using them to classify cervical cancer from Pap smear images.
Pap smears are a standard test for cervical cancer. How is AI being used to improve that process?
Cytotechnologists usually look at the images, trying to find abnormal cells that could be cancer, but that takes time and it can be subjective. Deep learning models are being trained to do this automatically. They can see patterns in the images that humans might miss.
Like a second set of eyes, but much more accurate and faster.
Exactly. This could lead to earlier detection of cervical cancer, which means better outcomes for patients.
Another example of AI improving healthcare.
And it shows how deep learning could revolutionize medical imaging in general.
Let's talk about one of the biggest problems in healthcare today, Alzheimer's disease.
Alzheimer's is a terrible disease that affects so many people. One article talks about using bioinformatics and machine learning to find new targets for treatment.
Therapeutic targets, what are those?
It's a specific molecule or pathway that plays a part in the disease developing. If researchers can find these targets, they can develop drugs or therapies that interfere with those pathways and slow down or even stop the disease.
Like finding the weak spot and creating a weapon to target it.
Exactly. With Alzheimer's, researchers are using bioinformatics and machine learning to analyze huge amounts of data, genetics, brain imaging, everything, looking for these targets.
Like a high-tech detective, looking for clues to figure out what causes Alzheimer's.
Good analogy. With more data available and better computational tools, we're getting closer to understanding this disease and finding ways to treat it.
That's good news. It's encouraging.
It is. Shows how bioinformatics could help us find new drugs and give hope to patients with these really tough diseases.
Now let's shift to another big problem. Antibiotic resistance.
Antibiotic resistance is a huge concern for public health. It could make it much harder to treat bacterial infections. One article talks about fragment-optimized chalcone derivatives. They target the OMP-A protein in Acinetobacter baumannii, which is resistant to multiple drugs. It could be a new treatment approach.
Acinetobacter baumannii, that sounds dangerous.
It is. It's a type of bacteria that can cause lots of infections. Pneumonia, urinary tract infections, bloodstream infections, and it's becoming resistant to a lot of antibiotics, so it's tough to treat.
So how do these chalcone derivatives help fight it?
Chalcones are compounds that are found in plants. Researchers have been looking at them as antibacterial agents. They tweaked the structure of these chalcones to create derivatives that target a specific protein in Acinetobacter baumannii called UMPA.
OMP, what does that do?
It's an outer membrane protein that's important for the bacteria to survive. If you target this protein, you can weaken the bacteria's defenses and make it easier to kill with antibiotics.
It's like designing a special weapon that targets the weak spot.
Exactly. It's a promising new way to deal with antibiotic resistance. We need new strategies to stay ahead of the curve.
To wrap up our AI and bioinformatics discussion, let's look at an example of how simple comparisons can be powerful.
Right. One article compares different deep transfer learning models. They're trying to classify cervical cancer from pap smear images. The goal is to figure out which model is the best at detecting cervical cancer.
Like a competition to see which AI model performs the best.
That's it. And by comparing them, researchers can see what works well and what doesn't. This helps them create even better AI tools for healthcare.
So progress in AI comes from working together and competing.
Yeah.
Everyone's trying to come up with the best solution.
And that's what makes it so exciting. There's always a push to be better, which leads to these breakthroughs that could really change healthcare.
We covered a lot in this segment. AI vulnerabilities, personalized medicine, the fight against antibiotic resistance. AI is definitely becoming a bigger part of bioinformatics, driving innovation in many different areas.
And in the last part of our deep dive, we'll look at some of the bigger trends and challenges facing bioinformatics.
Welcome back to the deep dive. We've covered so much, RNA sequencing, AI, you name it. It's clear bioinformatics is changing healthcare and research, wouldn't you say?
Oh, absolutely. It's constantly evolving, new discoveries all the time. Makes it hard to keep up.
That's exactly why we're here. We explored those breakthroughs, but now let's zoom out a bit. What are the big trends, the challenges shaping bioinformatics?
Data. The sheer amount of it.
More than ever before.
Yeah.
Genomic sequencing, health records, even data from wearables. It's like a tidal wave.
It is a tidal wave of data. And that's both exciting and a bit daunting. We've got all this info to study, to develop new treatments, but how do we manage it all, analyze it, make sense of it?
It's one thing to store it, but we need to get insights from it.
That's where bioinformatics comes in. We need better algorithms, models, visualization tools to cut through the noise and find what's meaningful.
Like finding a needle in a haystack.
Another trend, the blurring of lines between disciplines. Bioinformatics isn't just biology or computer science anymore. It's everything. Math, stats, physics, chemistry, engineering.
A real melting pot of science.
It has to be to tackle the big problems, like understanding the brain, personalized medicine, antibiotic resistance.
Collaboration is key, isn't it? Experts working together, bringing different perspectives.
Absolutely. And it's happening more and more, partly because of open science, data sharing.
Open science, that's all about sharing data, methods, research freely. Breaking down those walls.
It speeds things up. No more reinventing the wheel. We can build on each other's work, move faster.
Like a global team of scientists, all working on the same puzzle.
Another thing, bioinformatics is becoming more accessible. Tools and technologies that used to be limited to a few experts are now available to many.
Cloud computing, that's a big part of it, right?
Yeah.
Easier to access computing power, analyze those big data sets.
Yes, and online platforms are making it more user-friendly, so you don't need to be a computer whiz to do complex analysis.
It's opening doors for new researchers, even in places without a lot of resources. Exciting to see.
It is. But there are challenges, of course. One of the biggest is finding enough trained people.
The demand for bioinformaticians is huge. People who understand both biology and computer science to develop those algorithms, analyze the data, and explain what it all means.
It's hard for universities and training programs to keep up. We need more investment in education and training to prepare for the future.
And not just the traditional science backgrounds. We need diversity in bioinformatics.
Absolutely. Another challenge, making sure this technology is used ethically. As we gather more data about individuals, privacy and bias become big issues.
We need clear guidelines, regulations to ensure it's used for good, and doesn't just make existing problems worse.
It's not simple. Scientists, ethicists, policymakers, the public, everyone needs to be talking about this.
Can't ignore it. The future of bioinformatics hinges on using these tools responsibly.
It does. We've seen some amazing things today. Cutting-edge sequencing, AI transforming healthcare. It's a field with so much potential, always changing. But we need to stay mindful of those challenges.
Collaboration, innovation, responsibility. Those are key to moving forward. And that wraps up our deep dive into the world of bioinformatics. Hopefully you've learned something and gotten a glimpse of what the future holds. Thanks for joining us.
Thanks for having me. It's been a great discussion.